everyone. Thank you for joining me this evening. I am a little under the weather. I got COVID running through my family right now,
Oh,
so unfortunately, but I'm going to make it through this. I hope.
you just got COVID.
Yeah,
Sorry, man.
I know, I know. Um, so, uh, my name is Jay Kilian. I work at Maryland Department of Natural Resources Resource Assessment Service, Monitoring and Non-Title Assessment Division. That's the official where I am. I've worked on stream and river health assessment since 2001 when I started at DNR. I work under the umbrella project called the Maryland Biological Stream Survey, and we are all over the state assessing the health of our waters. In doing that, how we do that, we look at water quality and physical habitat quality and biological quality. We assess the health of the streams based on what's living there. And so, in doing our work over the years, we have built quite an inventory of our aquatic biodiversity. And we work very closely with our natural heritage program at DNR, who oversee all rare, threatened, and endangered species. We work closely with them. We collect data on all the aquatics and we provide that to them, and we work with them to assess the status of what is rare, what needs protection, and so forth. This black-banded sunfish is one of my favorites, but we've done a lot of work on it. It is by far the rarest fish in Maryland at this point in time. And I know many of you probably have heard about the Maryland darter, but this is, which is probably at this point, well, uh, probably, maybe not probably, maybe is extinct at this point. But this fish, black-banded sunfish, is near and dear to my heart. It's one we've worked on. It's still here, and it is very rare. All right, so you can see the black-banded sunfish screen, right? Everybody can see my screen.
Yep, we're all good.
I just switched to. Okay, I just want to make sure you can see what I'm seeing. Okay.
Yep.
All right, so just to give you a little bit of background, the number of freshwater fish species in Maryland, I have on that slide 80 to 100, and I know a lot of people will be like, well, why can't you be more precise? But there's a lot of fish that use freshwater, more than a couple dozen that use freshwaters on occasion, and so it's hard to draw the line when you say freshwater fish. What's the diversity? But they use there's many species that use freshwaters on occasion, migratory and so forth. So the best I can do is 80 to 100 in terms of our overall diversity. But when you look at the conservation need, it's around 29 species right now currently listed as species either rare, as either threatened and endangered, or just in need of conservation. They're on the watch list of those 80 to 100. And then I have the list down at the bottom of 6 species that we know are gone. And most of those are extirpated. 5 of those are extirpated, meaning they live in nearby states, but they're gone from Maryland waters. They once lived in Maryland waters, but they're now gone. The only one that is extinct is the Maryland darter. So I'm assuming this crowd knows extirpated is just gone from Maryland, gone from a watershed, gone from Maryland, but extinct is gone from the earth. And as you know, Maryland darter, known from only 3 sites up in Harford County, Maryland, presumed extinct at this point. Now, of those 6, trout perch at the bottom there, that disappeared from the Potomac River in 1911. That was the last time that species was seen. The rest of them were last seen within the last 50 years. So this is not something that happened a long time ago. This is something that happened in most of our lifetimes. And when people think about biodiversity loss, they think about they think about coral reefs and rainforests far away. But we are having local extirpation, local biodiversity loss in Maryland, and the other 5 species that you see there disappeared within the past 50 years, within our lifetime. This is not something that happened a long time ago. It's happening now, and it's happening with other species too. So I just want to emphasize that we are experiencing biodiversity loss. All right, let me. Okay, so black-banded sunfish. This is a beautiful. It's. I'm biased, but it's a beautiful species, and it's a member. It's a sunfish, so it's a member of the same family, Centrarchidae, of the same family as a smallmouth bass or a largemouth bass or a bluegill. But the black-banded sunfish is a member of a genus of that family called Enneacanthus, and they are very small. They are there's 3 members to this genus: the blue spotted sunfish, banded sunfish, and black-banded sunfish, and they all reach a maximum size of probably. I mean, I always use the term silver dollar. Most of us on this call know what silver dollar. So if I said that to my son, he would be like, "What? I don't even know what a silver dollar is." You know what I mean? But we're talking small. We're talking 3 to 4 inches max length. All 3 of the species that are in this genus are alive in Maryland. The blue spotted sunfish, they're all on the coastal plain of Maryland. So east of 95, coastal plain, and then Delmarva Peninsula. So the blue spotted sunfish is very common. The banded sunfish is less common, but you can find it. The black banded is the most rare of these 3 species in this genus, and they all are very small, and they all tend to be associated with acidic environments. And one thing, you guys are reading that slide, hopefully, but the bottom bullet there, they're tolerant of low pH, but they can spawn anywhere. So the thing about black-banded sunfish is they tend to be found in acidic environments, but amateur aquarists have spawned them in neutral pH. I mean, they're not particular, they're flexible in terms of their life history. Okay, so a really beautiful sunfish. Okay, so here's the distribution of the black-banded sunfish. Historically, it goes from northern New Jersey down to Florida, and that map is a little misleading because it almost looks like it's a continuous distribution. They are very disjunct in when you go and survey for them in all of these states. They are not continuous. They're found in patches. So that map doesn't really do it very good justice. They are habitat specialists. They're disjunct. They have been shown to they've declined in many states, including Maryland. They're considered extirpated from Pennsylvania, southeastern PA, in the coastal plain of Pennsylvania. Excuse me. And they are now considered state rare or vulnerable in a number of states, and vulnerable by the American Fisheries Society. They are not federally listed at this point, but they are on the radar screen of many states, including Maryland, because they're not doing so well across the range. Here is a map of the historical distribution of black bandeds in Maryland and Delaware, 35 localities as we know it. And so they were distributed, or they were found in the Chesapeake Bay drainage of Maryland and Delaware, but also the Delaware Bay drainage and also the Atlantic coastal bays drainage historically. To my knowledge, in Virginia, they are not known in the Chesapeake Bay drainage. They are known in the Chewan and the drainages way southeastern Virginia. So they're not in the Chesapeake Bay drainage of Virginia. So looking at the historical distribution in the Chesapeake Bay drainage, we had 19 locations in Maryland and Delaware, 12 in Delaware, and 7 Maryland records, not many. And you look at the records that I have listed there, 7 locations, and you'll notice they're all lakes, mill ponds. Most of them, at least, are lakes or mill ponds. So of the 19 Chesapeake Bay Maryland and Delaware historical records, 15 are from lentic habitats, pond habitats. Only 4 are from lotic habitats or streams. And in fact, there's only 1 record from the Pocomoke, and that is from a stream, not a pond or mill pond or lentic habitat. So why is this? I don't know. I mean, we don't know, but we have hypotheses, right? So here's our hypotheses. Most of Delmarva back in the day looked like this: swampy bottomland habitat, bottomland wetland habitats. And over time, we converted this to this. We ditched many of our streams and to drain them to make the land suitable for agriculture. But in doing that, we altered the aquatic habitats extensively, as you can imagine. We also, at the same time, removed beaver, which were pervasive on the landscape and were likely produced habitats conducive to blackbanded sunfish. And as this is all occurring, we're also building mill ponds. We're building mill ponds throughout Maryland, not just the coastal plain, but certainly in Delmarva, we built a lot of mill ponds. And so many of our records of blackbanded sunfish come from mill ponds that are still around today. And we assume that these habitats served as refuge habitats for blackbandeds over time. So in 2006, working with our natural heritage program, they were interested in, this is a rare species, and what's the status of the species? And so we set out to survey all of the historic Maryland localities where blackbanded sunfish were found historically. We also ventured into Delaware too, and we were working with Delaware to try to figure out, try to assess the species. We went to all 7 localities of where they were known to be in Maryland, and we threw everything. So I'm going to go back to this slide. So that's a boat electrofisher right there that we use. But we were throwing every gear you can imagine, seines, traps. I mean, we were doing everything we can. Now, it's hard to do a survey for rare species because they're rare. So you have to try to catch them with whatever gear you can. And we were trying everything we could to try to find them so that we could confidently say, this species is not in this habitat anymore, So, and it's hard to do because they're rare, and you're trying to detect a needle in a haystack in a way. So we were throwing everything at them, not just this boat shocker, but everything we could. And what we found, and we've surveyed multiple times over the years, these 7 localities, but we did find them. We know they're in Idlewild Wildlife Management Area, which is an area off of the Nanticoke. We surveyed the other historical locations, and we haven't been able to find them. So Idlewild, I mean, it's a cool place. It's a weird place. It's located just outside of Denton, and it's located along the Marshio Creek, which is an amazing, beautiful creek. And it's an old sand and gravel quarry. It's been dug out, and as they dug, they're called borrow pits. They dug and then they filled with water, and there's dozens and dozens of these ponds within the floodplain of the Marshio Creek. Black-banded sunfish are located within some of these, and some of the ponds are directly connected to the marships, others are not. It's a weird mix of habitats, but they're not in all of them. They're in some of them. So what we learned through our surveys, and we did surveys of mostly of Maryland, all of the Maryland historical localities, a lot of the Delaware historical localities within the Chesapeake drainage, and then also Delaware localities within the Delaware River drainage. And what we saw through all of our surveys, and again, we're throwing everything at them that we can to try to find them because they're hard. We found that the species that has declined across Delmarva in general. They're extant at only 5 of 23 localities surveyed thus far. And the pattern, as I'll get into in just a second, they're found in a pattern emerged in terms of when you find them, there is a pattern there. And so it got to the point where when we were surveying for them, we could go in there with whatever gear we were using, and right away, based on almost the community of fish that we were collecting at that time, we could say, yeah, they're not going to be here. Black-banded sunfish, to our knowledge, so far, they are very much associated with low pH, very acidic waters, less than 6, low ANC, low dissolved organic carbon. High dissolved organic carbon. These areas are tannic, black water. I mean, they are swampy areas. A very high abundance of aquatic and emergent vegetation, and the biggest thing is that the areas where you find them, you tend to find a more native fish community. You don't find bluegill. You can find them, but they're not in the abundance that you find elsewhere. Bluegill, largemouth bass, black crappie, these are all introduced species. They're the sunfish that everyone catches and loves. But when you find them in high abundance, you don't find black-banded sunfish. When you find black-banded sunfish, you find them with other native species, blue-spotted sunfish, mud sunfish. It's pretty stark in terms of the difference when you're surveying for black-banded. So what else have we learned? Looking at surveying what we know and we have, extant populations, and this is not just in Maryland, this is in Delaware as well. The remaining populations on Delmarva Peninsula are small. I mean, we don't have where we find them. They are not locally abundant. They are few and far between. I mean, we rejoice when we pick up a seine and there's a black-banded sunfish, and we're like, woo! I mean, it's not. I mean, it's not. They're rare. They're not abundant. Extant populations throughout Delmarva, both in Chesapeake Bay drainage and in Delaware drainage, are isolated. There's very limited potential for dispersal, genetic exchange, recolonization, et cetera, which doesn't pan well for long-term survival. And what we've learned through some graduate research of Maryland population is that there is low survivorship. It's things are not looking good. They're isolated, and there's limited exchange of genetics and limited chance for recolonization. So they're vulnerable. The Maryland population is very vulnerable. So why is it? What would happen? Well, it's the usual culprits, right? But habitat loss, stream channelization, like I said earlier, the removal of forested lands on Delmarva, channelizing streams. Hello? You guys still there? Can you still hear me?
Yes.
Okay, good. So it's channelizing streams, clearing of forests, and what we've done over time is we've removed the forested lands, changed the water quality. So we've changed the swampy habitats to more neutral water quality, which has also made for more suitable habitat for a lot of non-native species that we have introduced. So black crappie, bluegill, largemouth bass, and so forth, which compete and are predators of blackbanded sunfish. So it's been a kind of a combination of issues that have likely reduced blackbanded sunfish across our landscape. I have there at the bottom collection by aquarists that, blackbanded sunfish is a really beautiful species, and it has been collected over time by amateur aquarists that put them in their aquariums and so forth. I don't know if that's not really it's not probably the reason for their decline. It might be a threat potentially to the last remaining populations we have. So again, we started our surveys in 2006. 2007, based on our surveys of the Maryland populations, we immediately elevated the state status from threatened to endangered. We formed a conservation group with Delaware. We had Delaware, we had Maryland biologists, we had Nature Conservancy biologists. We had a whole group, and we developed a conservation action strategy for Maryland and Delaware in 2008, and have modified it since, which was a great step. And identifying what we need to do, what steps we need to take. In 2009 and through 2011, we funded a graduate research project to conduct life history and population size estimates on the Maryland population, which gave us a lot of important information. In 2015 and 2017, we collected fin clips from Delaware and Maryland populations, and we sent those. So we funded a genetics assessment. We sent those fin clips to Tanya Darden down in South Carolina, who is one of the experts on blackbanded sunfish, and she ran it with a lot of other genetics throughout the range from New Jersey to Florida, and gave us really depressing information. She, it was good information, but basically Maryland populations are so genetically inbred right now. They're almost clonal, like individuals are almost clones of each other. It's bad. Highly vulnerable to just blinking out. The Maryland population, as we know it, is not looking good, and we need to do something. But the great thing that came out of that genetic assessment was that we've identified that New Jersey, up in the pinelands of New Jersey, where populations are really robust, we learned that that population is the closest genetically to Maryland. So if we're gonna save this species in Maryland, New Jersey is where we got to go, based on that genetic assessment. So in 2017, the Conservation Action Strategy Group recognized that, yeah, we need to do something. What we have in Maryland is vulnerable as can be, because it's genetically stale and just depauperate, and it's vulnerable because there's no chance for recolonization. I mean, we need to do something in order to save what we know is in the only known population in Maryland. So we knew we needed to do some kind of captive propagation. We needed to do some kind of stocking in essence. So we recognized that in 2017, and we developed a captive propagation, translocation, and stocking plan. And in addition, through all our surveys over the years, we identified suitable habitats in Maryland for augmentation. Augmentation meaning adding to populations that are there, meaning Idlewild, and introduction, new populations. And the good news is that there is suitable habitat out there, especially for an experimental population, where we can learn, we can propagate and introduce and learn how to do it. And there's an area that we've called Wade Savannah Pond Complex down along the Nanticoke that it's on protected land. It has ideal habitat. It has the native fish community. It has no non-native piscivores. It's really ideal and a great place to wet our feet in terms of propagating these things or translocating them, meaning bringing them from New Jersey down to Maryland and releasing them, and learning how to stock these things and get new populations on the ground. So in 2018, based on the genetic assessment with South Carolina, we did identify that New Jersey pinelands populations are the most genetically similar to Maryland and suitable for a propagation translocation type of project. We went up and we worked with New Jersey wildlife biologists and did population estimates on ponds up there, because the one thing we didn't want to do is go up there and take sunfish from them and hurt their populations. But what we found with our population estimates is they have a ton of sunfish and can support a propagation and translocation project with us. And then the greatest thing is we partner with National Aquarium in Baltimore to initiate a pilot propagation project where we would grab sunfish from New Jersey, bring them back to National Aquarium, and try to propagate them for stocking. And so here's a timeline. I don't expect you all to read every last bit of this, but the gist of it is we started in 2018 and we ran into problems. These are sunfish. We brought them in and we had the initial collection. All the sunfish died because of a ranavirus. We collected additional ones in 2019 and didn't have much luck. And then COVID shut us down for the following years. It just hasn't been. We haven't had much luck, and these, National Aquarium, these folks they can bring a seahorse from the South China Sea into Baltimore and raise it and propagate it. So I mean, it's these folks are experts, and they ran into trouble. So it was frustrating for us. It was frustrating for them. We're still on this. We're still trying. We have collections planned for 2020 for this year in September. National Aquarium has bought and they've installed certain things to increase the likelihood of survival. And they're frustrated. I mean, because raising black-banded sunfish shouldn't be that hard. It's just we've just run into some bad luck. So we're very optimistic that this fall we're going to have some success. So we're moving forward with propagation and translocation over the next few years. Translocation meaning moving fish directly from New Jersey ponds right to Maryland ponds rather than trying to propagate them. We're doing both over the next few years, hopefully. So what else have we learned? So black-banded sunfish are very hard to detect. They're rare. They're locally not abundant. So it's hard to find any rare species, period. And like I said, we've already thrown a ton of gear at them trying to be confident in when we when we say they're not here, we want to be confident in saying they're not here, and it's not the result of us not using the right gear, etc. So there is something, and many of you have probably heard of this. It's environmental DNA. It's a very promising technique that's being used and more and widely. But basically, it's detecting the species DNA within the environment by just taking a sample of water. And the idea is that a black-banded sunfish swimming through a pond or a stream or wherever is peeing. It's shedding cells. It's excreting DNA into the environment, as every species does. And the idea is you collect water samples. You have to have a species-specific primer to detect, to detect that species. And then you run it through a PCR, and if it's there, you'll find it, right? I mean, it seems pretty easy and obvious. If the DNA is there, great, you got it. It's there. The species is there. And it's being used widely. It's very less expensive. I mean, it's very expensive to throw a crew into a stream and try to find a species. It's way less expensive to go out there and just collect a water sample. The cons of this approach is there are many unknowns affecting the reliability of this approach. It's still young, and we've worked on this for black-banded specifically, and we haven't had much luck. It's a great tool. It'll get there. We just haven't gotten there. So in 2021, we partnered with USGS Leetown Science Center, and we did a number of things. We've used filtration. There's a number of methods of I'm not going to get into the details of it, but filtration, flocculation. There's a number of ways to try to extract DNA from a water sample so that you can detect the species that are there. And there's seasonality to it. We've done a ton of things, and we've tried. And again, here's another timeline. But the gist of it at the end is it hasn't been very positive. There's been mixed results. It hasn't been very good. I wish it was so much better than this. There are a number of reasons why we haven't been able to. We're testing the eDNA tool, but what we're showing is that it's not reliable to go out and use to detect whether or not black-banded sunfish are present or absent. We haven't given up. We've tried a number of different techniques, passive eDNA samplers, different types of filters. You know, we've tried it, and it's not working yet. I'm not giving up, because this is a really awesome approach, because it's so less expensive than taking a 4-man crew out and trying to find a species. But it's not working yet. So moving forward, we're gonna continue to work with USGS on refining an eDNA method, hoping that it pans out. Of course, we're going to continue working with National Aquarium of Baltimore. I hope you all keep your fingers crossed. We're going to collect in September, and I hope we can get a captive population in National Aquarium and propagate them, so that we can start to augment the population in Idlewild and then reintroduce them elsewhere. In 2025, we plan to reassess the Maryland population and hope to initiate translocation, as I said, direct movement of individuals from New Jersey down to Maryland without the propagation part. So we're trying to attack this, you know, at all angles, if that makes sense. So that's it. Are there any questions?
All right. Yeah, Jay, can you unshare, come back and turn
Yes.
on your camera, and then we'll do questions and answers. I'll put a spotlight on you.
Am I unshared at this point?
Yeah, you're unshared, but we can't see your face.
Okay. Huh. How can I don't know how to get my face back.
Okay.
Might be better for all of us.
I know, amen.
All right. Well, at the bottom it says stop video, but there's a little carrot, or anyway, there's but let's go. Clark asked, thank you so much, Jake. That was fascinating and sad and hopeful, and all the everything rolled up into one. Clark has a question. Has the species ever been bred in captivity?
Yes, that's a great question. In fact, it's been bred by a number of aquarists, even in Maryland. It's not a hard species. That's what's so mind-boggling a little bit. But it's been bred by amateur aquarists easily, not only at pHs less than 6, but up to 7, like literally within your living room aquarium. So it's not that hard of a species to breed in captivity. It's just that's why National Aquarium is so frustrated, because it shouldn't be this hard, and we shouldn't have run into this trouble. So I'm hoping that from this point forward it's going to be easy. But no, it should be easy. It's a sunfish. It's not that hard to breed, and there's a lot of literature out there that describes how to breed it. I mean, it's not that hard. It shouldn't be that hard.
Following on to that, BS Brian asked if the aquarium is able to figure it out, the captive breeding, would it be possible in a program like the Sunfish program already in some Maryland schools?
Oh, that would be awesome. Yeah, I haven't thought that far ahead, but that would be fantastic. Yes. Okay, so there's raising of sunfish in Maryland schools, and trout. They do that at trout in Maryland schools, and then they stock them. I would love it to get to the point where we're raising black-banded sunfish in elementary schools and releasing them. That would be fantastic. But maybe someday, soon.
Katie asks, what is their role, they are being the fish's role in the Maryland ecosystem?
Well, that's a good question. I mean, certainly they fill the role of native sunfish, eating insect larvae and of course being prey up by other larger fishes. No specific. I wouldn't say that we're losing. Like the ecosystem, with them gone, the ecosystem won't collapse, of course. But I mean, they serve an important role in a blackwater stream river system, where predominated by native species. I don't know if I'm doing this justice, but you know, there's no specific. It's like if they disappear, it's the ecosystem will collapse, but we don't want them to disappear. You know what I mean? So,
right.
I'm sorry.
And I was going to ask about the eDNA stuff because Matt Ogburn will be doing a talk for us on eDNA. Yes. So come back for that. That's one of our. I'm fascinated with this new technology, but then you mentioned that it's not working right for this species. But Karen asks, has eDNA methods been successfully developed for similar fish species and similar in size, etc.?
Yeah, eDNA. And Matt, I know Matt. He's pushing the science. And it'll be a great talk. There are certain environments that are more conducive to eDNA. What we're finding with our eDNA work with blackwater, highly acidic environments is that it's harder to detect when they're there. And eDNA is really promising. I mean, I literally, it would be awesome just to go out and grab a water sample and know exactly everything's there and in the abundance that they are. And I mean, it would put me probably out of work, but it would be awesome to be able to do that. But there's a lot of problems with it. And one of them is the acidic environment, the blackwater systems, and what we're finding with black-banded. It's not that eDNA isn't out there; it's detecting it and with reliability. And the acidic environment, the DNA will break down quickly in acid situations. You also have the high tannins in high organics in the areas where you find black-banded, and we think that is getting in the way of being able to capture and detect the DNA that's there. So there's a lot of I mean, the science is young still, and Matt's doing a ton of work on it, and he's having success, but not in black-banded, and not in blackwater systems. He's looking at as to my knowledge, he's looking at shad and herring and migratory species, and he's having some success. Based on our work so far in these acidic environments, high tannic environments, the eDNA is not really helping us yet. I hope it does eventually, but we need more work.
Randy asked a question that I kind of had. You mentioned that the Maryland population was homogeneous and not genetically diverse, yet we're going to New Jersey to get specimens that are similar. So it seems kind of conflicting, genetically similar. So could you explain that?
Yeah, that's a great question. We are based on our genetic analysis, we are living with a Maryland population that is doomed. There's no flex, there's no adaptability. The genetic diversity is nothing in Maryland, but the closest, based on the range wide genetic analysis that Tanya Darden did is that Maryland and Delaware populations are most closely related to New Jersey. Far more diverse right now. So the idea is, if we don't do anything, the Maryland population is doomed because it has no adaptability because it's genetically clonal. The closest relative is New Jersey, which has really robust populations. Let's bring some of that diversity down and reinfuse the Maryland population with some viability, some diversity, which equates to viability. And that's, I mean, it's like that's where we are right now in terms of our goal is to build the genetic diversity of Maryland. But I understand the question that, you know, we are inbred, but our closest relative, quote unquote, is New Jersey. And I think and infusing more diversity into our Maryland population will go a long way to helping the species. Does that answer the question?
I think so.
All right. I hope so.
Yeah. So we have Jim. Do you want to explain your question? Unmute.
Hey, Jay.
Hey.
You stated that the fish likes low pH, high tannic water, but on that first historical map that you posted, it showed an area really far north, like up around the upper Susquehanna. And there's kind of like no really tannic water up in that area that I know of. So was that like from a long time ago that they were found in that area? And then secondly, the most tannic water that I've ever seen or experienced was in the Nanticoke. And I was just curious as to whether they have ever been located down there. And also, how does the water increase in water temperature affect their breeding and population?
That's a lot of questions, Jim.
Sorry, man.
I don't know about the conditions of that upper Susquehanna record. I do know that though you find them where we find them still extant in natural environments, they're in highly tannic area. I think, but they can spawn in circumneutral regular waters that are far more common throughout Maryland than the tannic ones. But I think what we are thinking is that when you get to where they can compete against the non-native species, meaning largemouth, bluegill, black crappie, etc. Those non-native species do not reproduce well in acidic waters, and I think that's where the black-banded kind of has its foothold, and they can, because the other non-natives are not doing well in that, they can reproduce well in that. They can have success. So, but that's not to say that if you throw them in a pond that is pH of 7, they'll do just fine too. Provided that there's no competitors or predators. So it's interesting. What were the other questions? I'm sorry. I should have written them down.
That's all right. So the other one was about the Nanticoke and also about water temperature.
The Nanticoke is where the extant Maryland population is, and it has some great habitat. In fact, it has areas where we think we can reintroduce them. It has suitable habitat where we think is a great area to reintroduce them. So the Nanticoke's the focal area right now. And what was the last one? Temperature. Temperature.
Water temperature.
I don't know. I mean, these are warm water fishes. I don't know how much climate change is going to affect them, to be honest. I mean, there are other species in Maryland that are going to be more affected by warming temperatures. I think I'm not too concerned about climate change on black-banded sunfish, but but we don't know everything about them. But there's other things that we should be concerned about.
Thanks.
Does that make sense?
Yep, sure does. Thank you very much.
Going on this, Nita asks about, are there any blackwater habitats, low pH tannic waters on the western shore that are potentially suitable? And were they historically more widespread beyond Delmarva? And then again, the stressors, is it the predation by non-native fish the greatest single threat?
That's a great question. There are low pH tannic waters on the western shore of Chesapeake Bay, especially down in Charles County and St. Mary's County, Mattawoman Creek, Zakaya. Those creeks are seemingly suitable, but we have no records historically of black bandeds there. Why? There's you could talk for days in terms of why and why not, but no records of them down there, but there is suitable habitat down there for sure.
To follow up on that, does the conservation strategy include the identification of these habitat restoration opportunities with potential stocking?
It does. It does. We've identified several that right now we're looking at. I mean, there's a lot of potential. If we were to get a captive propagation or translocation program rolling, which we're hoping to do, there's a lot of potential habitat out there. I think what we're focusing on is to do it to augment our current population where we know it, Idlewild, and then to introduce them and kind of as a pilot into the other location that I mentioned, Wade Savannah, along the Nanticoke, to try to just learn, to just kind of tiptoe into stocking these things. And this would be ponds protected with a native community, protected land. What does it take to get them propagated? How well do they do? And then learn from that, and then move to the beyond that. I hope I live to see it. The stocking them elsewhere and getting more robust populations in our riverine systems and so forth. But we're kind of baby stepping at this point, and we haven't had much luck. So I'm hoping for better luck, and with the National Aquarium and with everything, and getting to a point where we have so many sunfish we don't know what to do with them. That's the plan.
Hey, Jen asks about snakeheads. Do snakeheads tolerate low pH, and have they expanded into marshio?
Yes, they're all throughout marshio. So that's just another. And they're also within the ponds in Idlewild where we find black-banded. So yeah, snakehead, blue catfish. The non-native invasives are, they're they are what they are. But yes, they found their way, and they're a factor in the whole conservation of them as well.
Henry.
Yeah, go ahead.
Yeah. Well, Henry asks, what are the other states that are doing in terms of conservation or restoration of the populations that are experiencing the same issues?
As far as I know, Delaware's worked with us. They're not doing much now. They've had some personnel changes over the years, but they've been very active with us on black-banded sunfish. But there's not much going on there. I think what we're doing in Maryland will help them. Virginia has done some genetic assessments back in the day, and they have more robust populations. Of course, they're not in the Chesapeake Bay drainage, but they're further south. And New Jersey is very, New Jersey has the pine lands of New Jersey is, in terms of mid-Atlantic, is the stronghold for black-banded sunfish. I mean, you go up there and they have, I've pulled seines in Maryland and Delaware a billion times. And if you find one sunfish, you're like, whoo, it's amazing. You go up to New Jersey, you pull one seine and there's so many sunfish. So they've got robust populations. And what's great about New Jersey is that they are helping us. They know they realize that they have what they have, and they are working with us and allowing us to bring individual sunfish from their populations to Maryland. So I think Maryland and Delaware are the ones that are, in terms of populations, suffering the most. Virginia has a better population. New Jersey has a ton of them. So regionally, everyone knows the importance of this species, and so I can't what's the word? I can't. New Jersey, they've been really supportive, and we need them in order to bring back our native population. They've been really supportive. So,
Jay, when the historic records that you had, were those locations on privately held land or were they publicly held land?
Probably a mixture of both. Mill ponds are in Delaware are mostly publicly accessible. No, it's a mix. It's a mix. But I will say that anywhere we've surveyed, we've reached out and gotten permission by the private landowner.
And there hasn't been any pushback from the private landowners.
No, we with our statewide survey, not black-banded related, we most of our work is on private land, and yes, we get no's and we get yeses, but in general, well, I don't know if I can say most, but we only access where we can access. But many landowners are pretty supportive.
That's good to hear. Do we have any other questions for Jay this evening? Is there anything that we can do to help the black-banded sunfish?
I wish. I wish. Maybe we'll get so many propagated out of National Aquarium that we'll need help holding them until we can release them. But right now, no. But just interest in clean water and habitat protection. That's what everyone needs to do. I mean, this is the rarest fish in Maryland right now, and there's many more that are on the brink, of imperilment, and it's just a matter of keeping active within your own watershed, and what's going on, and knowing how, and being active there. I think that's not for black banded specifically, but for all the rare fishes and everything else. I think that's the most important thing.
Yeah, and it's important just to realize that these, the extinctions are happening near and far away and near, and happening
Yes.
right under our noses. So we want to do our best to keep the pieces that we have in the puzzle. Yeah. Thank you, Jay, so much for sharing your time, fighting through your illness or not feeling good, and most importantly, for all the work that you're doing to help not only the black-banded sunfish, but monitor and keep our streams healthy, and so that we know what to do. So we really appreciate all that you're doing out there for us.
Oh, well, thank you. Thank you for being interested in what we do. I appreciate it. I appreciate everybody's time.
Thanks, Jay. We
All right.
appreciate it.
Yeah, thanks, and stay well, stay curious, stay outside. We'll see you next week. We'll mind the gap, learn all about gap analysis. See y'all soon.
Take care.
Good night.
Good night.